people of him wrote in Wikipedia that he is known for unorthodox methods. What is right? So probably we will see now some unorthodox approach to genomic DNA and interesting linguistic analogy of baby talks. So enjoy it.
Thank you. Since we all have some attachment to biology, we also know what is phylogeny and what is ontogeny. And in individual development from fertilized egg to the adult individual, we see all the evolutionary way. We see fish, we start with amoeba, then something like sea cucumber, then you see the fish, and so on. In other words, individual development of the organism essentially repeats the evolutionary path from the most ancient evolutionary times to the organism of today. Similar thing we actually observe in development of language. There are plenty of theories of origin of language, but there is one which is very appealing, and this is baby talk theory. Whatever language you take, the babies pronounce first very simple sounds. And I think that perhaps in majority of languages of different groups, mama means mama, kaka means kaka, pipi means pipi. And these are those sounds which babies pronounce first. Of course, they babble all kinds of sounds, but the adults who do know language react, of course, to the right ones. And this way the baby gets feedback and starts to use those simple words already consciously. So the first words in the talk of a baby are very simple, repetitive words. So since I heard most of babies in my life Russians, so the examples which I give are mostly Russian, but some of these examples are international. Like I was explained recently that the baby who wants to drink in Vietnamese says something like wong wong. Ham ham, Vietnamese, clear what is it. The simplest are mama, papa, baba, pipi, kaka, sisi, bobo, and so forth. And there are variations. Slowly, the baby gets to more sophisticated words, but mostly in the beginning, they all are repetitive. I just continue to next page. Now, many of these sounds are simply imitations of sounds from nature, like copying geese, ga ga ga, copying clocks, tick tock, frog, quack quack quack, and so forth. The car, BB. Interestingly, that imitations of a rooster by adults, babies cannot pronounce that. They are different in different languages. Kirikoko, you know. Kukariku in Russian. And unbelievable cock-a-doodle-doo in English. Well, chmok chmok is, of course, imitation of kisses. But say skirly skirly, wooden leg from a Russian fairy tale, surely this is by adults. So essentially, adults also talk very much by repeating words. The sounds copied by children are brilliantly illustrated by the piece from Mark Twain about Tom Sawyer, when his friend was showing the ship steamer docking to the pier and pronouncing all those sounds like ting-ling-a-ling. This is clearly the bell of captain. Then the machine would do choo, 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 and at the end, or something like that. It's a vapor getting out from engines going to stop. So these are adult forms. Little babies cannot say x, 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 x. It's difficult for babies. But adults do that. And they're using the repeating words. And these are all examples from different languages. Like in Ukrainian tilki-tilki, it's just barely fit, tilki-tilki. Or tipa-tipa in Hebrew, just a little bit, tipa-tipa. Trochi-trochi, the same in Ukrainian. Clearly that baby cannot pronounce that. But adults do. So adults, they also speaking simple repeats. These are other examples. Well, interesting English, teeny teeny. The single word is pronounced not teeny, tiny. But two words together, they pronounce teeny teeny, because it's more convenient to repeat this way. Tsatsa. Tsatsa, it's a girl showing up. Again, the baby cannot pronounce it. It's an adult word. Zip, 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 calling chicken. Well, the 5 years old, 4 years old, of course, would be able to say zip, zip, zip. Then these words started to change. From ideal repeats, they started to mutate. And here are the examples of those mutations. Of course, mommy instead of just mama. Baby instead of baby, written B-E-B-E. Interesting that there is a fruit called papaya you know about. In Brazil, there is another similar fruit, but much larger. And of course, it's called mamaya. You know well, surprise word, oh la la, in French. Here is a nice word, Totoye, it's Aliska, daughter of Misha. Instead of Tsotoye, she says Totoye, turning adults' language into simple repeats, which are more convenient for babies. You recognize Russian word balalaika, very clear where from it comes. From repetition, slightly changed repetition. Same tarataika. Tarataika is a sound produced by the cart with wooden wheels riding on the rocks. You could use instead something like tram tarataika, imitating the same sounds. Chinese yin-yang, a philosophical combination. Here's an interesting word, su-sukat, imitate baby. Tsap-tsarap, catch about cats. Here is a Latin, willy-nilly or volens-nolens, against will. And here is Mongolian, olgoy-hor-hoy, which is a fairy tale creature. And here is nice Russian word, nazhuzhukalся, about a drunk man. And finally, futi laptignuti. It's already a verse. And the verses, a poetry, they are repetitious. And people use it very broadly. Songs, verses, repeating, especially refrains, repeating. There are more examples like that. Tap-lap is lousy work in Russian. That you put it something not really checking, tap-lap, and made something. Drum tararam, it's a mess, just almost showing a sound of something ruining. The same trach tararach, it's imitating the thunderstorm. Bayu, bayushki, bayu, lullaby. And famous, of course, counting rhymes for seek and hide game. It happened that I had, when I was a schoolboy, I had a little sister and we had a nanny, and it happened that the nanny was German. So when she heard us saying "Ene bene rech kentermenter zech, ene bene raba kentermenter java," then you go and look who is hiding where. And she said right away, "No, it's not like that." She said, "This is ine mine minke tinke fader rode rolke tolke wigel wagel weg." Clearly sounds in German, right? But it's also very repetitive. Another Russian combination of that purpose, eniki beniki yeli vareniki, eniki beniki klots. But that goes on and on and on. Human speech is very repetitive. This is a piece from a very frequent citation to Martin Luther King. In his speech, he said that if you want to say that I was a drum major means in the orchestra, the main drum. Say that I was drum major for justice. Say that I was drum major for peace. I was drum major for righteousness. And when they recently put a monument to Martin Luther in Washington, they wrote on it, I was a drum major for justice, for peace, for righteousness. And immediately there were protests that he was misquoted. And indeed, when you repeat something, it adds additional expression to what you say. And it appeals more to the listeners. That's why, for example, in church, very frequently, you see sermons with repetitions. Repetitions intentionally made to reach the righteousness ear. Human languages, quite likely, originated from simple, repetitive words, continued with their mutated forms. And even today, the languages operate with simple repeats, mutated forms of repeats, and longer tandem or dispersed repeats, refrains, like in songs. And what I want to say today, that exactly the same can be said about biological sequences. That is, they started with simple repeats, which then changed, mutated, so that today we often do not recognize in those sequences original simple repeats. If you take human genome, 3 billion letters, and make a full vocabulary of all the words, in this case, words of length 15 letters. Total amount of different words of that size, purely combinatorially, is about 1 billion. In other words, we would expect that every word of the full spectrum would be repeated in human genome 3 times in average. It's not the case. It turned out that some words are repeated not 3 times, but more than 1 million times. Repetition of TTTs, repetition of AAAs. Then you see pretty frequent repetition of TG, 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 AC, AC, AC. Again, GT, 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 which is the same CACA. Then you go to ta, 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 and so forth. In other words, in the ranked list of all 15-letter long words in human genome, on the top, you see repetitive words. Now this one, although it doesn't look like simple repetition, it is also repetition. It's so-called allo sequence. Allo sequences are about 300 nucleotide long sequences, which are repeated in human genome every 2,000–3,000 nucleotides. It's a sort of, I don't know, pages, numbers of pages, or whatever you call it. And their function is not clear. Nobody knows what they do there. But there's so much that even they get to the very top of the frequent words. So this sequence is a part of allo, and this is complementary sequence also from allo. So one would say that the simple repeats are a major component of human genome. If you have 1 million of TTT, 1 million of AAA, then somebody is missing in the list. And indeed, of 1 billion of possible words, 300,000 words are not there. You don't find them. That is, there are 15-letter words which are not present in human genome. These are bad words. Something wrong about these words. They completely avoid it. The most known, well-known type of repeats is those pathologically expanding aggressive classes of triplets. GCU class, GCC class, and GAA class. They cause neurodegenerative diseases and chromosome fragility, and the mechanism they do it is you originally have, say, in norm 20–30 repeats like that, and then all of a sudden they expand to several thousand of repeats sometimes. And that accompanies or causes the disease. One important thing to say at this moment is that if you have repeating GCU, 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 GC, UGC, it's the same like repeating UGC, as well as repeating CUG. And if you take complementary strand of that duplex encoding this GCU, then there you find AGC as well as GCA repeating and CAG. In other words, such duplex could be represented by 6 different ways. And in the beginning, when these diseases were first detected, the doctors who would see the sequence repeating, they would read the sequence with the first letter of the repeat. And each time it was different. There were 6 different ways to call the same repeat. They thought that these are 6 different repeats. These are not. And actually, it was 1907 when my friend Thomas Betekin, in Mini Minky Tinky Father Rudy Rolki Tolki, he wrote to me, what is going on with those diseases? They are called differently, but I see that they are the same. It's all GCU or GCC or GAA. There are only 3 types. And what is special about this? Why they expand? What is going on with them? So studying DNA curvature, DNA packing in nucleosome, I tried to find out what is special about these sequences. I couldn't. But in a series of exchanges by telephone with Thomas, we all of a sudden developed the following very provocative thought. We said, if these sequences are so crazy that they could expand arbitrarily, despite the fact that they are inside of well-protected, well-developed Homo sapiens, and surely it would be stupid to allow these things to happen. And yet, Homo sapiens, with all its organization, is helpless against these aggressive repeats. What if these repeats were present in earliest days of evolution, at the days of origin of life? Perhaps there were other sequences around, but these will surely win. Because they are crazy, they are aggressive, they will win, they will take all resources on them, only they will multiply. This way we came to the idea that the sequences like GCU, 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 or GCC, 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 and complementary GGC, they probably were the very first coding sequences. The life started with these sequences. This boiled down to a whole new theory of early molecular evolution. Here is one important chapter of this theory. The point is that we could figure out that the very first triplets were GGC, GCC, for example. But what would be the next triplets to come? Again, common sense suggests that the next triplets to come would be the same triplets but mutated in one position. So first, then, after them, there would be, instead of GGC, there would be GAC. Or instead of GGG would be, well, GGC would turn into GGA, GGG, GGU, so you have point mutations. Similarly, complementary ones, those which make a complementary strand, also would mutate, so that instead of GCC, you would also have GCG, GCU, GCA. And this way, we're going from one triplet to another, going through all the triplets, but in what order? The order is suggested by chronological order of appearance of amino acids in evolution. It is true that the very first amino acids which appeared in evolution were glycine, alanine, and a little bit of aspartic and valine, and they were synthesized abiotically. It's just pure chemistry. You don't do anything. You don't have biology at all around, and you simply, because of thunderstorms and lightnings, you get in the primordial atmosphere of the Earth these amino acids. This was first imitated by Stanley Miller in 1953. He actually synthesized this way 10 different amino acids. But in 1953, because of poor analytical tools, he only could detect alanine, glycine, aspartic, and valine. And later on, in say 1987, already all 10 amino acids could be detected. So how to determine this order of amino acids, how they appear? The valine, that is glycine and alanine, it's clear. They are highest yield in abiotic system. But what about other amino acids? And there are plenty of various considerations which should be the first amino acids, considering chemistry, atmosphere of the Earth, conditions, considering sometimes mathematical symmetries, all kinds of theories, all kinds of suggestions. So I collected all these suggestions, total amount of 101 in December of 2007. And I averaged these suggestions. That is, I designed from this data an average. That is, how many of these suggestions would insist that alanine was before glycine. So, and I would put glycine rather, because the votes say no, glycine was earlier. Actually, they were simultaneous. So the order of amino acids shown here is a result of analysis of 100 some different criteria, which are all speculations. So the result also would be speculation. However, a remarkable thing happened. If you put to these amino acids those triplets which encode them today and put them in thermodynamic order, in the order of thermostability, you see that they go the same order as those amino acids. This diagonal shows that amino acids appeared in the order of appearance of their corresponding triplets. Or better to say, triplets appeared according to their thermostability. GCC, GGC is the most stable triplet. The least stable triplet is UUU, AAA, for phenylalanine and leucine. And this correspondence is just shocking, because as soon as we noticed that, the whole table was built in no time. Indeed, if you take GC, it mutates to GAC, one-point mutation. And what amino acid would be used by this mutation? Clearly, aspartic. And aspartic was third in the series. Simultaneously complementary would appear, which is GUC. GUC could be made from GCC by mutation in the middle. And again, question is, which would be the next amino acid? Just next in a row. Next is valine. So forth, by the same way, like you mutate GGC to GGA, these are lines with descending thermostability. So the next thermostable is GGA, complementary is UCC. GGA is the same for glycine. But what is UCC? We take all the list of remaining amino acid and see which is the first. The first is serine in this chronology. So the chronology is perfectly following thermodynamics. And thermodynamics, that doesn't necessarily mean that the life started in hot conditions. Simply, thermostability is important in any conditions. So from this evolutionary table of triplets, several important consequences were formulated and confirmed by sequence analysis. And I wouldn't go in details now. I only make a point about origin of life. Darwin, in his writings, never formulated what is life. But he was pretty prolific in descriptions of what is natural selection. And here is one of these descriptions. Here, if variations useful to any organic being ever do occur, assuredly individuals thus characterized will have the best chance, blah, 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 produce offspring similarly characterized. Rephrasing, putting it in a shorter form, individuals with useful variations will self-reproduce. And I thought that this is not bad for definition of life. The life, following Darwin, would be self-reproduction and variation. After all, what is the most important in life according to Darwin? Natural selection. So formulating natural selection, we, in a way, formulate what is life, self-reproduction and variation. Of more than 200 different definitions of life given by various philosophers, naturalists, the closest to that is definition by Oparin, another giant of evolution. Any system capable of replication and mutation is alive. Again, self-reproduction and variation. And then let's return to the codon evolution chart. If we take the very beginning of that chart, the line GCC, GGC, this would be self-reproduction. No changes here in the first line. It's just copying itself and copying itself. If you want to have life, you have to introduce changes. And the changes follow right away. The point mutations in those triplets to follow. In other words, if you take the very corner of the codon evolution chart, then you say glycine-alanine encoded by GGC-GCC duplex. It is not life yet, because it's only self-reproduction. But as soon as you introduce mutations in those triplets, you introduce variations, and then you get self-reproduction with variations, and this is life. So unexpectedly, from this reconstruction of evolution of first amino acids and first triplets, we came to a definition of life. Tentative definition. Because we didn't witness that life, we cannot say that life is that. But it follows logically from whatever we built so far. Life is self-reproduction and variations. Then I did the thing which really caused an explosion in the ranks of philosophers. I said, there is no definition of life. There are 200 some definitions of life, which means there is no definition of life. They would rather agree that there are 200 of them, and one of them is theirs own, and there is definition of life. So each one would insist on his own definition of life. And to put them together and discuss, to get logically to the definition would be impossible because of ambitions, of individual ambitions of these people. But I did the same thing which I did with order of amino acids. I didn't ask for arguments. I just put everything together, giving it equal weight. Of course, some are more stupid, some are more clever, but I don't know. So I gave them all equal weight. And I simply counted which words they use to get their definitions. And these are these words. You easily could group them, and these groups are shown here. So I took 123 known definitions of life, not 200. Why? Because 123 are published in 2 collections of definitions, and all others are scattered. If I would pick those which are scattered, somebody will tell you, aha, you picked up those which you like. No, I like them all. So I took those which are tabulated, and here is life, the thing which is defined. And of course, when you say living or alive or being or biological, these are all related words describing life. And they are all used, and the whole group makes 199 related words describing the life, with root life or similar roots. But also there is a system and related words like organism, order, network, organized, together making 155 words among those definitions. Matter, chemical, complexity, reproduction, evolution, environment, energy, and ability. You now could put all these words together and design the definition. And I actually wrote such definition, including all these major categories. And when I sent this paper to Origin of Life journal, the critics was, well, all the rest what you say, reproduction with variation, it's all bullshit, it's speculation. Leave that long thing. But I didn't like that long thing because it contains things which do not really belong to definitions. They belong to life, but they don't belong to definition, like energy. Of course, there couldn't be life without energy. But this is commonplace. It's not an essence of life. Essence of life is not energy. And probably essence of life is not chemistry. Because one could imagine, in principle, replicating entity with variations, which is not made of chemistry, like in computer or something, or some mental constructions. So when you put these categories together and try to select among them, select those which would make the core of definition. Well, mathematical is the best definition. The right definition is the one which is both necessary and sufficient. And any combination you take from these doesn't make it, except for pair, reproduction, and evolution. These two categories appear to be both necessary and sufficient for the definition of life. But this is exactly what we've got from the codon evolution chart. Reproduction with changes, reproduction with variations. So we again have the same definition. Life is self-reproduction with variations. If you open Google on Trifonov definition of life, you see how mad people are with that definition. One of the most interesting reactions amongst others was, this is completely wrong paper, but very interesting. And the issue of journal Biomolecular Structure and Dynamics, where I finally published that paper, it wasn't accepted by Journal Theoretical Biology, with all reasoning like that. You are wrong because you're wrong. Origin of Life, I decided not to waste time, send it to Journal Biomolecular Structure and Dynamics, and the chief editor of that journal is, well, I wouldn't conceal the fact he's my friend, and I just wanted to make a shortcut. And when I sent the paper to him, I said, but be careful with that paper. It's sort of controversial. And next day I got the answer from him, controversial, it's outrageous. So he said, I will publish your paper, but only after 19 commentaries to your paper will appear as well. And I got 19 articles commenting on that definition, of which 15 were strongly negative, but 4 were just positive. Now returning to human genome. When in 2001 the first version of human genome was published, they also published the percentages of different types of sequences in the genome. You already know that protein coding and RNA coding together make about 3% only of the genome. All the rest is so-called non-coding DNA, of which simple sequence repeats make 3%. But this is underestimated, because if you take T, yes, it's simple repeat. But what if you have T, it's also simple repeat, slightly compromised. So therefore, this is underestimated. Transposable elements make 45% of a whole genome. It's a scandal. 45% of human genome. Human, it's top of universe. 45% stupid sequences. So the Lander and his co-authors concluded that repeat sequences account for at least 50% and probably much more of the human genome. Now, why probably much more? Because you I think, know that the human genome is not one continuous sequence. It's not even 20-some continuous chromosome sequences. Within each chromosome, you have something like 300 pieces, contigs. They are placed in the right order, but in between, people don't know what is the sequence in between. They cannot sequence through these gaps, because these gaps are simple repeats, and nobody knows how many are there. Now, I had a student, Eliezer Aharonovsky, who once made the following analysis. He said, what if those ancient amino acids are still repeating in some sequences in a large amount? Then we look what are the sequences and could make conclusion about their age. And somehow he started with human protein coding sequences. And after he fused them together, different exons together to get continuous sequences, he discovered that he actually has a separate collection of first exons. And he took first exons of human genome and made an analysis on frequencies of amino acids. First, it was amino acids frequencies analyzed. If you remember now those three aggressive triplets, three types of aggressive triplets, total 18 different triplets. These 18 different triplets, they encode these amino acids, leucine, alanine, glycine, and so forth. And the corresponding triplets are these shown here. These are all from aggressive triplets. So when Eliezer analyzed how many repeating aggressive amino acids he has in the proteins, in the first exons of humans, he got almost all of them on the top of the list. Here is the scores, how many you find them. Only two are off, phenylalanine and cysteine. By the way, these are pretty rare amino acids. All right, then we took eukaryotic sequences in general, not just first exons, all exons, and we got similar result. We got that about 75% of all repeats in three letters, 3 leucines, 3 alanines, and 3 lysines. About 75% are aggressive amino acids. In prokaryotes, even more, 89% are aggressive triplets. What is going on? The proteins are made of aggressive triplets. I mean messenger RNAs made of aggressive triplets, encoding those aggressive amino acids. Could it be that protein sequences actually are all originally made from the aggressive repetitions? And we don't see all the original repeats just because they have extensively mutated. We still have an excess of those aggressive things, but not 100%. They all mutated. So if this is correct view, then what we would expect? First of all, if we take large ensemble of messenger RNA sequences, so the first thing we would see there is ideal repetition of some codons. We don't know which ones, all of them or not, but we expect ideal repetition. If they started with repetitions, then the repetitions we should see. Not long repetition, not the whole length of messenger, of course, because that would be nonsense, but patches of repeating sequences. Now, if you see the patches, you don't see continuous repetition. How you recognize the former repetition, which is now in the sequence? Everything mutated in between. You get your triplets, and between them you get different triplets. But here's an interesting prediction. If the sequences originated from certain triplet which was continuously repeating over the range, and then it mutated, then between two triplets, identical triplets, the middle one, the sandwich structure, the middle one would be most likely a first derivative of that triplet. That is point mutation of that triplet. This way, the sequences would still remember where from they started. So the codons sandwich between two identical codons should be their point mutation derivatives. This is a very strong prediction. Because if you take codon usage, for example, then overall codon usage, the most frequent codon is AAA for lysine. So then you would expect AAA coming into middle of the sandwich. No. We'll see soon. So prediction is that not AAA, but first derivatives would be there, because after all, we expect that the memory is still there. And finally, one more expectation, that those codons, which are more often in tandem repeats, should be also of higher usage in non-repeats. That is, we don't have repeats anymore. We throw away repeats. We see only non-repetitive parts. And make a codon usage. And which codons are on the top? Those which are in repetitive parts, the most active. In other words, those single codons, non-repetitive anymore, they originated from multiplying, expanding codons. Although the repeat you already don't see. So we, therefore, undertook the analysis of the largest non-redundant database of messenger RNAs available, of total 5 billion codons. From eukaryotes, prokaryotes, viruses, organelles, all together. After all, origin of life is common for all of them. And that is published just last year. Here are the results of the analysis. Am I right? Yeah? So these are results of the analysis. We looked for, of course, these are protein coding sequences. We were interested primarily in triplets, in codons. So there are 60 4 different triplets. But of course, termination triplets do not repeat. They do not appear in the protein coding sequences. Therefore, there are 61 types of repeats. They are split in 10 different families. Family of GCC aggressive triplets, GCU aggressive triplets, alphabetically GCA, and GAA aggressive triplets, and also 7 more families like that. Here they are grouped in the families, and total occurrence of repeats, at least 3 repeating codons together, is tabulated here, and what you see that these aggressive families are on the top of the list exactly as expected. And the rest is going down, 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 down. The least repetitive is ACU family with those triplets shown here. In other words, tandem repeats of all 61 different codons are observed, strongest for aggressive groups, as expected. Now take a sandwich. I bring you example with a sandwich GCU, ABC, GCU. Total number of such sandwiches is 3 million. And this is which ABCs we find in the middle of the sandwich. First of all, we find the same triplet as we started with. So if you have 2 neighboring triplets separated by another one, then in the middle you most likely find that your triplet. But if not, then you find point mutations: GU, GAU, GUU, GCA, GCC, and only GAA intruded this list. It's at the top of the list, by the way. It's 200, 100,000, while at the bottom there are barely 20,000 at the very bottom. So GAA is the topmost in non-repeating codon usage. And yet it didn't make it on the top. We would expect that they would be on the top here. And next topmost in codon usage is AAA. Also, it didn't make it to the middle. On the top, you see first derivatives of GCU. And the same thing is observed for most of other codons. I wouldn't say for all, but for most. That is, this is a general rule. There is a memory of tandemly repeating sequences of triplets. Almost every triplet tandemly repeating has in the middle frequently its point mutation. Now take thick sandwiches. Put in between several triplets. So the effect which I demonstrated to you relates to this point. This point, if you have repeating XYZ and XYZ and single triplet in between, then most likely you would have it the same XYZ in the middle. But then if you take thicker sandwich, 2 triplets inside, 3 triplets inside, up to 100 triplets inside, you still see that XYZ dominates. It doesn't go to the 0 background. Same with first derivatives. These are all first derivatives of XYZ. They are highest when the sandwich is thin, with 1 triplet between. But no matter how many triplets between them, you still have these in excess. In other words, in the sequence where there is no repeats anymore, you find identical triplets at long distance from one another. They belong to formerly continuous repetition of that triplet. This is illustrated by these 2 graphs. So the first derivatives between the identical codons in messenger RNA keep memory of initial tandem repetition of these codons. So that the sequences like XYZ, NNN, NNN, XYZ, NNN, NNN, XYZ are likely descendants of continuously repeating XYZ. So the sequences still keep a memory about from what they started. Now, if you just check how many different triplets you have in a given gene, if you take shuffled sequences, random sequences, then you have this red distribution, shuffled sequences. That is, sometimes you have as many as 10, 12, even 14, but when you take the same triplets in the protein, in the messenger. But when you take natural sequences, you very clearly see that the proteins like to have some of the triplets abundant, which in our language now would mean that they started with these triplets repeating. So we took proteins which are at this tail of the distribution, and just looked at them to check how these triplets are distributed there. And we got the following fantastic picture. This is just one example. We didn't try to get the best example. We even took a very ancient protein. This time is ribosomal protein. That is, that protein had a lot of time to mutate. And it didn't. It still remembers of what pieces it was made. You see those, we call them bricks. The red brick containing GAA triplet, GAA, 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 or point mutation of it, GCA, GTA, GAG, GAC, GCA. Then all of a sudden this picture changes and you see GCT, 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 and point mutations of this GCT. So the red brick is followed by green brick, then again by red brick, red brick, red brick. Frequent triplets make clusters, remnants of original ideal repeats. You could take any messenger RNA and do that trick with them, and you know right away which pieces originated from what. Of course, you have to then suffer through statistical proofs that this is not a random effect. But this is done easily if you know statistics. So our calculations indicated that at least 35% of genomic sequences, not only of messenger sequences, are statistically proven to originate from tandem repetitions. At least 35. That means, that doesn't mean necessarily that the rest is not never been repetitive. No, they've been repetitive. Simply they change so much that we cannot prove it by statistical means. We have other means to prove it. So, and then a third prediction. The third prediction said that if you have some triplet which likes to be repeated, and then you take non-repetitive part of those messengers and check whether this repeat is frequent there as well. And the answer is yes. Those which are more frequently found in tandems, they are also found more frequently in non-repeating regions. The dependence is dirty. I wouldn't deny it. After all, proteins have their own interests. They're fighting for their own choice of codons in amino acids. But this is all ripples on the background of major phenomenon. That is, the repetitiveness of the triplets shapes the codon usage table. It's not active genes, non-active genes, home genes, whatever. It is repetitiveness of those triplets which makes the codon usage table. It's best seen when you take the table itself. Here we separated the codon table in independent repertoires. Like alanine repertoire, of course, it's 4 amino acids, but arginine, for example, it's 6 amino acids and 2 different repertoires. Similarly with leucine, 2 different repertoires. So let's look at the repertoires. We have 2 columns of numbers opposite every triplet. First column is codons in non-repetitive part of messenger. And second column, repeats in repeat part of messengers. And then you see that the highest frequency codon in alanine is GCC on the top. And the highest repetitiveness codon is GCC. Arginine, both highest repetitive and highest codon. So there are exceptions, all right, I wouldn't deny it. 1, 2, 3, 4 exceptions. 17 of 21 codon repertoires have the most frequent codon, also the most repetitive. This result, of course, came as a surprise, because there are zillions of factors known to influence the codon usage. There is a whole discipline of codon usage. Plenty of literature on that subject. What we find is that more frequent codons keep memory of tandem repetition of these codons in the past. They are more frequent because they are very repetitive. The triplet expansion of codons is a major single factor shaping the codon usage. So now I remind you that according to theory of early molecular evolution, the very first genes have been repeats GGC, 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 and complementary GCC, 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 GCC. And life started with the replication and expansion and subsequent mutations of tandemly repeating triplets GGC and GCC, which is self-reproduction with variations. Life continued then to spontaneously emerge within the primitive early genomes and further on in form of replication and expansion and subsequent mutations of other tandem repeats as well. Again, self-reproduction with variation. In other words, life never stopped emerging. Here we're coming to a famous statement by Darwin. He said that if we could conceive in some warm little pond comes from this statement. In literature, you frequently find warm little pond of Darwin. It's from here. All sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed. He didn't know about nucleic acids at that time, so he meant actually life. Ready to undergo still more complex changes, at the present day, such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed. In other words, what Darwin actually said, that life cannot start again. It started only once. This was a unique event, and nothing similar happened later. According to the view which I am developing, the emerging life is not consumed by the earlier life, but rather protected by the environment within the cell, because that new life emerges inside of the genomes. perfectly protected in conditions much milder than at the moment of origin of original life. Now we're returning to classical junk DNA and selfish DNA. The Orgel and Crick and simultaneously Doolittle and Sapienza in 1980 said that the tandem repeats is a class of selfish DNA, living for themselves, so to say. Fortunately, not harming much. They're living for their own interest. They are actually more than just parasites tolerated by genome. They are even more than building material for genome. The building material was the idea of Susumu Ono, which he called junk DNA, not because it's a garbage DNA. He meant junk as a junkyard for old cars. From parts, old parts, you could make a new car. The tandem repeats represent constantly emerging life, and genomes are products of their everlasting domestication. They emerge in form of repeats, I would say stupid repeats, but then they start to mutate and something good happens. If bad, then they are selected against. But if something good happened, then they are acquired, domesticated, and stay in the genome. Genomes are built by the expansion and mutational domestication of the tandem repeats. Genomes are the repeats, some already unrecognizable. We actually could consider the tandem repeats, since they symbolize origin of life, as sort of living, simple living organisms. We call them simplicimus, in other contexts we call them preons, not prions, but preons, from the word pre, meaning before. For genomes, in this symbiosis, the accepted repeats are useful, but new repeats are dangerous. For repeats, genomes are natural habitats, but initiation is at high risk because genomes wouldn't tolerate, die out if the repeat happened in the wrong place. Wrong place means in important functional place. It will be a deadly mutation. So from here, on the basis of this completely new view of the genome and its evolution, we could make an important prediction, which is that genomes should be equipped by defense systems against constantly emerging repeats. We could even say that in case of triplet expansions, the aggressive triplet expansions, this defense systems fails. Now we return to those first exons. When we analyzed where along the first exon those repeats appear in human genome, then we discovered that they appear in the beginning of the exons. The amino acids repeats in prokaryotes at the bottom here are far less frequent compared to eukaryotes. Why is that? Are they not important in evolution of prokaryotes? Yes, they are important, but prokaryotes have a very brutal system of selection against the repeats. As soon as a repeat emerges in the wrong place, this individual dies out. The population doesn't die out. That individual dies out. They could victimize one individual. Elephants cannot victimize themselves, even individuals. Now, why it is at the end of the exon? Well, the idea is that probably this repeats at the end of the exon actually coming from nearby non-coding sequence. Non-coding sequences are very rich in repeats. So exon could acquire some of these non-coding sequences and turn them into exon sequences. But since they are rich in repeats, you of course acquire remnants of the repeats and see more of them in the beginning. Then we get an interesting idea about role of introns. Introns and non-coding sequences are depots of tandem repeats. And this is a defense system to push the tandem repeats away from exons into introns, save the coding sequences. And that's probably what happens when nature plays with alternative splicing. The alternatively spliced piece of exon either belongs to intron or belongs to exon. So it is a play around until the evolution will take decision whether to accept or to reject it completely. So defense in prokaryotes is simple: brutal negative selection, death of individuals contracting the repeats. Defense in eukaryotes, expulsion of the repeats into introns. This is speculation. Nobody so far said any words about systems of defense against tandem repetitions. Therefore, so far it's speculation. It has to be studied. What are possible defense devices? First of all, the expansion goes through slippage structure, formation of slippage structure. And in order to prevent the slippage, one way is to limit the space so that the slippage loop wouldn't have space to form. Then the space should be crowded, and nucleosomes may well play an important role in it. Now, slippage loop is formed, it's good to cut it out. So there should be enzymes specific for most aggressive repeats to cut the loops out. Now, repeats could be methylated. The GGC repeat contains CG repeating. GGC, next up again is GGC, so CG at the border. If you methylate this CG, then it stops expanding. And finally, there could be also sequence-specific nucleases which would recognize most unpleasant repetitions and simply cut them. The simplest life forms, simple tandem repeats, represent a whole class of pathological agents not considered as such up to now. The genomes evolved under constant attacks by various repeats. Apparently, most of the attacks are normally stopped by the defense systems. Some of the new expansions or insertions are accommodated by the genomes. Some are neither stopped nor accommodated, causing disaster. A different view on cancer, expansion diseases, and diseases with unknown causative agent. The repeats in those diseases are not symptoms. They are cause of the diseases. And here are those people who did the work. First in the list is Zohar Koren. It is Eliezer Aharonovsky. At one moment, he all of a sudden decided to change his name. My friend Zachary Frenkel, very witty guy, he said, aha, he probably running away from tax office. Thomas Betekin is now in München. At that time when we started this work, he was in Magdeburg. Misha Zemkova is here in the audience. So thank you very much.